Welcome to another deep dive, everybody. Today we are going full neurologist for you folks. We're diving into canine meningiomas.
Oh, yeah.
And we're dissecting a really cool correspondence article from Acta Neuropathologica.
Okay.
And it's titled, Canine meningiomas are comprised of 3 DNA methylation groups that resemble the molecular characteristics of human meningiomas. Quite a mouthful.
It is.
But very interesting stuff.
Yes, very much so. And, this research is really exciting because it's looking at those molecular level changes in canine meningiomas, and that has some really big implications for both our canine patients and potentially even human patients.
Definitely. So before we get into the study itself, I think it would be helpful to kind of define DNA methylation and why it even matters.
Sure. So DNA methylation is essentially a process that can change gene expression, so turning genes on or off, without actually changing the DNA sequence itself. So it's kind of like a molecular switch. It plays a really, really critical role in regulating how our genes are expressed.
Yeah.
And when this process goes wrong, it can contribute to some pretty serious diseases like cancer.
Okay, so that makes sense. So let's jump into the study now. What did the researchers actually do and what did they find?
So they used whole genome bisulfide sequencing, or WGBS, to look at DNA methylation patterns in 29 canine meningiomas.
Okay.
And then on top of that, they also did RNA sequencing to see which genes were actually being expressed in these tumors. So this gave them like a really comprehensive view of what was going on at the molecular level.
And what did that molecular landscape actually show?
Well, the big finding was that there are 3 distinct molecular groups of canine meningiomas, and this actually mirrors what we've seen in human meningiomas. Now, to come to this conclusion, they used a variety of clustering methods and dimensionality reduction techniques. They didn't just rely on one method, which actually strengthens their findings quite a bit.
So it's like using multiple tools to make sure you're on the right track.
Exactly. So they used k-means clustering, hierarchical clustering, UMP and TSNE, and all of these pointed to this 3-group structure.
Oh, wow. So they used different approaches just to confirm that these 3 groups were real.
Exactly.
Not just like artifacts
of
a specific analysis. Okay. So can you give us a visual sense of how these techniques work?
Sure. So imagine you have a giant data set with thousands of data points. And each of those data points represents a tumor. These techniques that they used, clustering and dimensionality reduction techniques, help us to organize and visualize this data.
So it's like taking a jumbled up puzzle.
Yes.
And sorting the pieces based on their shapes and colors.
Yeah.
So you can actually see the full picture.
Exactly. And if you look at figure 1 in the paper, you can see how they actually apply these techniques to the data.
Okay. So let's walk through those figures. I'm sure they have some pretty important information to show us. What's in figure 1A?
So figure 1A shows something called a consensus matrix, and this is based on the k-means clustering.
Okay.
So essentially, it demonstrates how the tumors are grouped together based on similarities in their DNA methylation profiles.
Okay.
Essentially, it's a heat map. Where each row and column represents a tumor sample, and the intensity of the color indicates how small the samples are. And you can actually see 3 distinct blocks of color in that figure.
Oh, wow.
Which represents our 3 molecular groups.
So tumors in the same group share a similar methylation pattern.
Yes.
And those in different groups have different patterns.
Exactly.
Got it. Okay. So figure 1B takes us a step further, right? And it shows the gap statistic, which helps determine the optimal number of clusters in a data set. So it's confirming that the 3 clusters which represent the 3 groups best fit the data.
Precisely.
Okay, so both that visual representation in the consensus matrix and that statistical analysis using the gap statistics support the idea that there are 3 distinct groups.
Right. And figures 1C and 1D actually provide even more evidence using something called the continuous distribution function, or CDF.
Okay.
And these graphs plot the cumulative probability distribution of a variable. In this case, it would be the similarity scores between the tumor samples and the jumps that we see in these curves. Further support that there are 3 distinct groups.
Gotcha. So I'm seeing a trend here.
Yeah.
Multiple methods all pointing to the same conclusion. What about figure 1M?
Okay. So figure 1E is a heat map that kind of zoom in on the specific methylation status of CpG sites, which are these regions in the DNA where a cytosine nucleotide is followed by a guanine nucleotide.
Gotcha.
And this heat map shows us the methylation levels of these CpG sites across all the tumor samples. And it shows us that each of the 3 groups has a distinct methylation pattern.
So it's like a molecular fingerprint that sets each group apart.
Exactly.
Cool. And lastly, figure 1F uses a dimensionality reduction technique called UMA.
Yes.
And this is a way to visualize complex high-dimensional data in a lower dimensional space, which makes it easier to see patterns.
Exactly.
So instead of trying to picture thousands of data points in a 1,000-dimensional space, which our brains can't really do,
right,
UAB projects this data onto a two-dimensional plot that we can actually understand.
Exactly. And in this plot, you can see the 3 distinct clusters, which again represent those 3 molecular groups of canine meningiomas.
Wow. So all 6 panels in figure 1, using all these different methods, strongly suggest that there are these 3 distinct groups.
That's right.
Of canine meningiomas based on their DNA methylation profiles.
Exactly.
That's really cool.
Yeah. And, these findings lay the foundation for understanding the biologic differences between the groups and how they might actually behave clinically.
And that's where things get really interesting. But before we get into that, I want to take a closer look at a specific feature highlighted in figure 1G.
Okay.
Hypomethylated regions, or HMRs. What are those and why are they important?
So HMRs are regions in the genome where methylation levels are actually lower than usual.
Okay.
So remember how we talked about DNA methylation being like a dimmer switch for gene expression?
Yes.
Well, in HMRs, those dimmer switches are turned up.
Okay.
Leading to potentially increased expression of the genes in those regions.
Got it. So less methylation in HMRs could translate to more activity in the genes they contain.
Precisely. And in this study, they found that group 2 canine meningiomas had way more HMRs than the other 2 groups.
So group 2 tumors have more of these turned up dimmer switches.
Yes.
Which could be a clue to why they're unique.
Exactly. It suggests that maybe gene expression in group 2 meningiomas is being dysregulated in a specific way that could contribute to their behavior.
So we've seen how these DNA methylation patterns can distinguish these 3 groups.
Yeah.
But what about the genes themselves? What did that RNA sequencing data reveal about the genes that are being expressed in each group?
That's where things get even more intriguing, I think. So the RNA sequencing data revealed that each group has distinct gene expression profiles.
Okay.
And these profiles actually have some really cool parallels to molecular groups that we see in human meningiomas.
Okay. I'm ready to get into these gene expression profiles. So let's start with group 3. What makes this group stand out?
So group 3 showed this really interesting enrichment of genes that are associated with the hedgehog signaling pathway.
Hedgehog signaling. I've heard of that. Remind me why it's important.
So the hedgehog pathway plays a really crucial role in embryonic development and cell growth.
Okay.
But when it's dysregulated in cancer, that can lead to uncontrolled cell proliferation.
Okay.
So it can be bad news. But the cool thing is that group 3 canine meningiomas actually mirror the Merlin intact group that we see in human meningiomas, which also show this enrichment of hedgehog target genes.
And Merlin intact human meningiomas generally have a better prognosis, right?
That's what we see, yeah. But, of course, more research needs to be done to confirm if this is true for dogs as well.
Yeah.
That's a crucial point. So group 3 is kind of like the good news group, at least based on what we know about human meningiomas. What about group 1? What does their gene expression profile tell us?
So group 1 shows enrichment of genes involved in inflammation, immune response, and IL-2 signaling pathways.
It's like their immune systems are in overdrive?
Yeah, you could think.
So these tumors are attracting a lot of immune cells?
Precisely.
Okay.
And this group looks a lot like the immune-enriched group in human meningiomas.
Okay.
Which also show this heightened immune activity. And in humans, those tend to have intermediate outcomes. They're not as good as Merlin intact, but not as aggressive as the hypermitotic group.
Gotcha. So group 1 falls somewhere in the middle.
In terms of prognosis. So let's talk about group 2 now, the one with all those HMRs. And that potential dysregulated gene expression, what's going on with their genes?
Group 2 showed this striking enrichment of FOXM1, which is a transcription factor that plays a key role in regulating the cell cycle.
Okay.
And they also had increased expression of cell cycle target genes, which suggests that they're probably proliferating faster.
So these tumors are basically primed for rapid growth and division.
Exactly.
Okay. And I remember you mentioned this group is similar to the hypermitotic group.
Yeah.
In human meningiomas, which are known for their aggressive behavior.
Yes.
So this FOXM1 enrichment and that increased expression of cell cycle genes, That's a red flag.
Yes.
Potentially meaning a more aggressive tumor. Is there any other evidence to support that?
Yeah. So they found a correlation between group 2 and a higher histological mitotic index.
Okay.
Which means that more cells were actively dividing in these tumors.
Oh, wow. Okay.
They also saw increased expression of MKI67 and TOP2A, which are markers for cell proliferation. Huh? And on top of that, group 2 tumors were more likely to be high grade.
Oh, wow.
So even further evidence of their aggressive nature.
So multiple lines of evidence point to group 2 being the most aggressive of the three.
It seems that way.
This is all really interesting from a purely scientific standpoint.
Yeah.
But let's zoom out for a second and consider the bigger picture. What does this actually mean for dogs and humans who are facing meningiomas? What are the potential implications for treatment?
That's the real question, isn't it? I think this opens up a lot of possibilities, especially for developing new therapies. So one key takeaway is that canine meningioma patients could actually be really valuable for preclinical pharmacological testing.
So what works for dogs could potentially work for humans.
Exactly.
And vice versa.
Imagine being able to test new drugs and therapies in our canine patients and then use that information to inform human clinical trials.
That could really accelerate the development of effective treatments for both species.
Precisely. And canine patients could even become invaluable participants in these clinical trials, leading to even faster breakthroughs.
Potentially saving countless lives.
Yeah.
Both human and canines. So this research has uncovered a lot about canine meningiomas, but it seems like there's still so much more to learn.
Definitely. One area that needs more investigation is whether there are molecular subgroups within these three main groups.
Okay.
Kind of like what we see in humans.
So it's like we've zoomed out and seen the big picture with these three groups.
Yeah.
But now we need to zoom back in.
Yes.
And see the finer details.
Exactly. And this will require even more sophisticated genomic analysis and potentially even single-cell sequencing.
Oh, wow.
To really unravel the intricacies of those subgroups.
And this deeper dive into these subgroups could be really crucial for developing targeted therapies, right?
Absolutely. Imagine being able to personalize treatment to each patient's tumor profile. That's like the ultimate goal of personalized medicine.
It's amazing to think that studying canine meningiomas could lead to so many breakthroughs for both human and canine health.
Yeah. It highlights how interconnected all species really are.
Yeah.
And the potential of comparative oncology.
Definitely.
Picking up where we left off, let's look more closely at the specifics of each of these molecular groups, starting with group 3, which we were calling the good news group.
Right, the one with that connection to the hedgehog pathway.
Exactly. So the hedgehog pathway, like we talked about, it plays a super important role in development and cell growth. And when that pathway is dysregulated, it's been implicated in a lot of different cancers, including meningiomas. And in human meningiomas, those that are classified as Merlin intact also have this enrichment of hedgehog target genes, and generally those have a more favorable prognosis.
So finding a similar group in canine meningiomas is potentially a good sign.
We hope so. Of course, more research needs to be done to confirm if the canine equivalent of that Merlin intact group also has that better prognosis, but it's definitely a good lead to follow. You know, imagine if we could develop targeted therapies that specifically address the dysregulation of that pathway.
That would be huge. Okay. Moving on to group 1, you mentioned their gene expression profile suggested a really active immune response.
Yes, group 1, similar to that immune-enriched group in human meningiomas, shows this really distinct upregulation of genes that are involved in inflammatory and immune processes, particularly those related to IL-2 signaling, which suggests a pretty robust immune response happening within the tumor microenvironment.
So their immune system is going after that tumor.
Exactly. And this raises some interesting questions about how this immune activity might influence tumor behavior and even the response to treatment.
That's a good point. Does a strong immune response always mean a better outcome?
Not necessarily. It can be really complex. You know, the immune system is there to help fight cancer, but sometimes those immune cells can end up promoting tumor growth or even make certain treatments less effective.
So it's not just about having an immune response, but understanding exactly how it's interacting with that tumor.
Exactly. And this complexity just highlights that we need to do more research to fully understand how the immune system is working in group 1 canine meningiomas and how we can actually use that to our advantage for therapy.
Okay, let's talk about group 2 now, the one with all the HMRs and that concerning gene expression.
Group 2, the one that's similar to the hypermitotic group in humans, it presents a much more aggressive molecular profile.
Can you explain a little more about what FOXM1 does?
Sure. So FXM1 is a transcription factor. It's like a master regulator of the cell cycle, basically. It controls the expression of all these different genes that are involved in cell division and proliferation. So when FOXM1 is overexpressed, like we saw in group 2 canine meningiomas, it can really push that uncontrolled cell growth, making the tumor much more aggressive.
And I remember you mentioned there was other evidence to support this aggressive nature beyond just the gene expression.
Yes, there are a few findings that back this up. So first, group 2 tumors had a higher histological mitotic index, meaning there were more cells actively dividing. And then they also had increased expression of MKI67 and TOP2A, which are very well-established markers of cell proliferation. And on top of that, group 2 tumors were just more likely to be high-grade, which is a classification that we usually associate with more aggressive behavior.
So we're seeing a lot of different factors all pointing to group 2 being the most aggressive of the three.
That seems to be the case, and this really emphasizes how we could potentially use molecular profiling to identify which dogs are at a higher risk for developing these more aggressive meningiomas. And early identification could lead to more proactive treatment strategies.
That makes sense. The sooner you know what you're dealing with, the better you can be at making decisions about treatment.
Yeah, exactly. And this research opens up some really interesting avenues for exploring more targeted therapies. Imagine developing drugs that could specifically inhibit the FOXM1 activity or disrupt the cell cycle pathways that are just super overactive in group 2 tumors.
That's a big question, isn't it? Can we translate these molecular insights into actually effective therapies?
That's the goal. And even though there's a lot more research to be done, this study is a really good starting point for exploring these potential therapeutic options.
Now, you mentioned before that human meningiomas could be further broken down into even more subgroups within those three main groups. Is there a possibility that something similar is happening in canine meningiomas?
That's a great question, and it's definitely possible this study focused on identifying those three big molecular groups, but further research with larger sample sizes and more advanced techniques like single-cell sequencing could definitely reveal these smaller, more subtle subgroups.
And what would be the advantage of identifying those?
Well, think about it this way. Identifying these subgroups could really change how we approach personalized treatment. Imagine being able to tailor treatment not just to the group, but to the specific subgroup, maximizing how well it works and minimizing those unwanted side effects.
So moving away from a one-size-fits-all approach to a more personalized treatment plan.
Exactly. And the technology is getting there. Single-cell sequencing is advancing so rapidly now that we can look at the gene expression profiles of each individual cell within the tumor, which could be really valuable in identifying these subgroups and understanding that complexity within meningiomas.
So it's like going from looking at an entire forest to being able to examine each tree individually.
Yeah, a great analogy. And beyond just identifying those subgroups, there are so many other research directions that we could explore. For example, looking at the proteomics of these tumors, which is the entire set of proteins that they express, could give us clues about diagnosis and treatment.
So not just the genes, but the actual functional molecules that they make.
Exactly. Proteomic analysis could show us unique protein signatures for each group, and that could lead to new biomarkers for early detection or for monitoring how a patient is responding to treatment.
And what about the tumor microenvironment? I know we talked a little about the immune response in group 1, but there's probably a lot more happening in that ecosystem around these tumors.
Absolutely. The tumor microenvironment, all the surrounding cells, blood vessels, extracellular matrix, they all play a huge role in how a tumor grows, progresses, and responds to therapy. So understanding how the microenvironment differs between these molecular groups could lead to completely new ways to intervene therapeutically.
So not just targeting the tumor cells, but also changing their surroundings.
Exactly. There's a lot of interest now in developing therapies that target the tumor microenvironment. So understanding its role in canine meningiomas could have implications for treating both dogs and humans.
Let's step back for a minute and think about what these findings mean from an evolutionary standpoint. What does it tell us that both dogs and humans have these distinct molecular groups when it comes to meningiomas?
It really points to this deep evolutionary conservation of these tumors across different species. The fact that dogs and humans separated by millions of years of evolution still show these similar molecular subtypes suggests that whatever mechanisms are driving meningioma development are very ancient and fundamental to how mammals work.
So it's not just a human disease or a canine disease. It's a biological process that goes beyond species.
Exactly. And this has a major impact on how we approach research into meningiomas. Studying these tumors in dogs can teach us a lot about the basic biology of these tumors, which can then be used to understand and treat human meningiomas.
It really shows how interconnected all living things are and how important comparative oncology is.
It does, and the ethical considerations that we've talked about really highlight how important it is to do research responsibly. If we're going to learn from our canine companions, we need to make sure we're taking care of their welfare throughout the entire process.
Absolutely informed consent from owners, carefully monitoring their well-being, minimizing any potential discomfort or stress, are all essential. These dogs are partners in this, not just subjects.
I agree. Their contributions should be recognized and respected, and they should always be able to participate voluntarily with their quality of life being the top priority.
This deep dive has been a really interesting journey into the world of canine meningiomas. We've talked about so much, from the details of the research to the implications for both canine and human health.
It's been great exploring this with you, and I hope everyone listening has learned more about how complex and important canine meningioma research is.
Welcome back to the deep dive.
Yeah.
So in this final part of our look at canine meningiomas, we'll be looking at where this research might lead us next and what other questions this study raises.
Yeah, this research is really just the start. It's opened up all these new paths to explore and highlighted how important canine models are for understanding and treating meningiomas, not just in dogs, but also in humans.
Let's talk about some of those paths forward. You mentioned identifying subgroups within those three main molecular groups.
Right. Think of it as we found these 3 continents, but each continent has all sorts of different landscapes and ecosystems. Within each of those 3 molecular groups, there might be subgroups with subtle differences, in their molecular profiles and how they behave.
So it's like zooming in even further on those UMP plots and finding even smaller clusters within the main ones.
Exactly. And figuring out those subgroups could be really important for developing even more targeted therapies. Imagine being able to tailor treatment not just to the main group, but to that specific subgroup so we can make it as effective as possible and minimize side effects.
That's the goal of personalized medicine, treating each patient as an individual.
Exactly. And, single-cell sequencing is getting so advanced now, we can actually analyze the gene expression profiles of individual cells in a tumor, which could be incredibly helpful for finding those subgroups and understanding just how diverse meningiomas can be.
So instead of looking at the whole forest, we're looking at each tree.
Exactly. And besides finding subgroups, there are tons of other research directions to go in, like studying the proteomics of these tumors. The entire set of proteins they make could give us more clues for diagnosis and treatment.
Looking at the actual molecules the genes are making.
Yeah, proteomic analysis could reveal these unique protein signatures for each group and lead to new biomarkers that we could use for early detection or to see how well a treatment is working.
We've talked about the immune response in group 1 tumors, but what about the whole tumor microenvironment? What's going on in that complex system surrounding those tumors?
The tumor microenvironment, all those cells, blood vessels, extracellular matrix, it all has a big impact on tumor growth, how it progresses and how it responds to therapy. So if we can understand how that microenvironment is different between those molecular groups, it could open up new ways to target the tumor.
So we wouldn't just target the tumor cells themselves, but also try to change the environment around them.
Exactly. And there's a lot of work being done to develop therapies that go after that tumor microenvironment. And understanding its role in canine meningiomas could be helpful for treating both dogs and humans.
Let's think about these findings from an evolutionary perspective. What does it mean that we see these same molecular groups in both dogs and humans when it comes to meningiomas?
It really shows how deeply conserved these tumors are across species. Dogs and humans are separated by millions of years of evolution, but we still see these similar molecular subtypes. So that suggests that the mechanisms behind meningioma development are really ancient and fundamental to how mammals work.
It's not just a human disease or a canine disease. It goes way beyond specific species.
Exactly. And that has a big impact on how we approach research into meningiomas. If we study these tumors in dogs, it can give us valuable information about the basic biology of these tumors, which can then be used to understand and treat human meningiomas too.
It's a great reminder of how connected all living things are and how important comparative oncology is.
It really is. And with all the ethical considerations we've talked about, it's so important to do this research responsibly. If we're learning from our canine companions, we have to make sure we're taking good care of them throughout the whole process.
Definitely. Things like getting informed consent from owners monitoring their well-being and making sure to minimize any discomfort are essential. These dogs are helping us out. They're not just subjects.
I completely agree. Their contributions to these advances should be acknowledged and respected, and they should always be able to participate voluntarily with their quality of life coming first.
This deep dive has been a really fascinating look at canine meningiomas. We've learned so much from the molecular details to the bigger implications for both dogs and humans.
It's been great talking about this with you, and I hope everyone listening now has a deeper understanding of how complex and important this area of research is.
I think they do, and this research has a lot of potential for improving the lives of both dogs and humans dealing with this disease as we wrap up this deep dive. I hope everyone stays curious, keeps learning, and continues to explore the amazing world of science from Vet Jar. Keep those minds-inspired hearts light and tails wagging.